Hi dear students, welcome back to one more session on chemical bonds and biomolecules, and let's continue with the next type of bonds called the hydrogen bonds. We were talking about the non-covalent bonds, and hydrogen bonds are one among them. And this mainly happens because of a sharing of a hydrogen atom by a covalently bound atom and another electronegative atom. Now, sometimes you can see that the hydrogen atom, now for example, if you look in this case, it is being linked towards, it is covalently bound to an atom called the oxygen. And this hydrogen atom itself can be shared by another electronegative atom. Now, if you have another oxygen molecule over here, it can be what? It can be shared. This hydrogen atom can be shared between the two oxygen atoms. Such a case, this is a case of what? Sharing of the hydrogen atoms happens, and that is a case of a hydrogen bond. Now, when you have when a hydrogen atom is being shared, it will have a hydrogen donor as well as a hydrogen acceptor will be present. Okay. Now, in this case here, the hydrogen donor is oxygen, into which the hydrogen is covalently bound, and the hydrogen acceptor here is oxygen. Now, in this case, you can see the hydrogen atom is being attached covalently linked to a nitrogen atom, and the acceptor is oxygen. So, the hydrogen donor and acceptor can either be oxygen or nitrogen, depending upon the case in which case the hydrogen is being shared. Now, in this particular case, in the last case, which is kept my arrow, you can see that the hydrogen atom is being covalently linked to one nitrogen atom, and that is being in turn shared to another nitrogen atom by a hydrogen bond. So, in the case of a hydrogen bond formation, what happens? You always have a what? A hydrogen donor as well as a hydrogen acceptor, and it could be either nitrogen or oxygen. In the case, either cases, it can be either oxygen or nitrogen. And hydrogen bonds are usually being formed between charged as well as the uncharged molecules. And what is the significance of this? We will just find out that. Like, these are the hydrogen donors and the hydrogen acceptors. It's clear. I'm not going to explain it much. So, hydrogen bonding can happen in different ways. Either it can occur in linear fashion, or sometimes hydrogen bonding, it can happen in a ring-like fashion, or sometimes in a dimeric fashion, or in some cases it can be also been found to be intramolecular. Okay, so in all the bonding cases where hydrogen bonding is happening, it is the hydrogen atom which is being shared between two different atoms, and the atoms are in such a way that they will be, one of them will be electronegative, highly electronegative. Okay, and either in chains, rings, dimers, intramolecular, or sometimes you can also find that in among the hydrogen bonds itself, okay, some of them will be stronger. Now, for example, hydrogen and fluorine. Fluorine is a very electronegative element, okay, highly electronegative element. So, a strong hydrogen bond can be formed over there. But oxygen and hydrogen, in the case of water, you have a weak hydrogen bond over there. So, compared to the covalent bonds, hydrogen bonds require less amount of energy to be broken down. But among the hydrogen bonds also, there are different types of bonds. Some of them will be stronger and some of them will be weaker, depending upon which electronegative atom is being involved in the bond formation. Now, as I had mentioned earlier, hydrogen bonding can be happening in between within the molecule itself, or it can be between different hydrogen molecules also. Now, in this case, you can see a hydrogen bonding is being what happening over here. This is one molecule. It is covalently linked to it, and it is hydrogen linked with oxygen, and you can find. But these are two different molecules. Just look here. This is a intermolecular hydrogen bonding. So either intermolecular, that is two different molecules can be interacted by hydrogen bonding, or within the molecule itself there will be intramolecular hydrogen bonds are also being present. Now, what is the relevance of this? Hydrogen bonding plays an important role in determining the 3D structures as well as the properties adopted by certain synthetic as well as natural proteins. Now, if you go to consider the carbon-carbon bonds, carbon-oxygen bonds, and the carbon-nitrogen bonds, which have been found in most of the polymers, the energy which has been needed to break a hydrogen bond has been found to be very much weaker. Almost only less, as much as 5% of energy is being needed to break a hydrogen bond. So, hydrogen bonds can be broken by a chemical or a mechanical means while retaining the basic structure of the polymer backbone. So, if you go to consider the hierarchy of the bond strengths, you have the covalent bonds. The covalent bonds are being stronger than the hydrogen bonds, and hydrogen bonds are stronger than that of the other non-covalent bonds like the van der Waals forces and all that. Now, let's explore where does hydrogen bonds happen within the biomolecules. In the case of DNA, we know that what? Base pairing, or in the double helical structure of DNA, nucleotide bases they follow the base pairing rule. The adenine pairs with the guanine, and adenine pairs with the thymine, and guanine pairs with the cytosine. And this is being carried out by using hydrogen bonds. A and T forms what? Two hydrogen bonds between each other, and G and C forms three hydrogen bonds with each other. And we know during the replication process also, this non-covalent hydrogen bond has to be broken down easily at each division of the zinc. That is being carried out with the help of the what? With the help of, because least amount of energy is needed for the breaking down of the hydrogen bond. Now, you might think about the ionic bonds which are happening during replication also. Ionic bonds are mainly being formed between the DNA and the histones. I'm talking about the hydrogen bond here, where guanine and cytosine, you know, these are what? Nucleotide bases which are being found within the nucleic acids. They are being linked together, or the base pairing is being carried out by using hydrogen bonds. Now, in the case of the protein structure also, what? Just like we have the what? You have the peptide bond which has been formed between the amino acids, and then after that the secondary structure. The primary structure has mainly been formed by the binding of different amino acids by peptide chain, but the secondary binding or the secondary bonding is mainly been formed with the help of certain what? Hydrogen bonding natures. Hydrogen bonds which are being formed. You know, the secondary structure forms alpha helix. When two chains are being rotated with each other in a helical fashion, you form a alpha helix. Or when they are being stacked one parallel to each other, you form a beta chain. Beta chain in the case of proteins. And for the maintenance of this secondary structure of the proteins, the hydrogen bonds do play an important role. And do keep in mind, the hydrogen bonds are of less energy compared to that of the covalent bonds. And if you go to consider what are the features of hydrogen bonds, always hydrogen bonds, they are being found to be highly directional. That is, in one particular, I told you, it is only from a hydrogen donor to a hydrogen acceptor. So the direction of the hydrogen bond plays a very important role to maintain the structure, and it has been found to be more specific than any other weak bonds, as they require a particular group that donates or accepts the hydrogen atoms. And the two other features, as we had already mentioned, is that the hydrogen bonds can be either intermolecular or intramolecular. Now, intermolecular is a one. Now, if you have two helical chains, between that, you can have a hydrogen bond. And in the case of energy, the covalent bonds have a higher energy, and beyond that you have the hydrogen bonds, and beyond that or lesser than that you have the van der Waals forces in the case of energy. So I hope this portion is clear to you. Thank you for now. We will continue with the other non-covalent bonds in the coming sessions.